Wow. Hey, welcome everybody to this fantastic thesis defense by Emily Moffitt, and I'm so thrilled to see all of you here and all of Emily's supporters and fan club. And I know she has a very large fan club because she is so incredible and has been doing so much with her thesis and in the department and actually in the region. So Emily came to us in 2013. She got her bachelor's at the University of Vermont, Bachelor of Science in Animal Sciences. And then you were a vet tech for a while. Was that here?
In California, yeah.
In California, okay. And then Emily joined the program, our master's program, in 2013. And it's really been such a joy to have you with us. I'm really sad that she's graduating, but I know you're going to do great things because you've already done great things here.
Thank you.
Emily has done a really amazing project looking at the stable isotope analysis of a particular songbird that needs some attention, as so many songbirds do during their migratory pathways. As she'll tell you, and during the migratory season, they're exposed to a lot of stresses. We need to know more about them. And Emily's work is going to show us some very interesting aspects of this bird's ecology during migration. So I also want to mention that Emily she works for SFBBO, San Francisco Bay Bird Observatory, that she has been an intern at the Santa Clara Valley Water District, that she has also been our student coordinator for our graduate program. Yes. All on top of doing a very, very demanding thesis. So I don't know where she gets the energy or the time, but she does it all incredibly well. So I wanted to acknowledge some of the people here today, so including her committee members. And actually we have Kristen Rugg, who is on Skype here from Oxford. And Scott, would you just introduce yourself?
Oh, Scott Schaefer, biology.
Great, thank you so much for being on Emily's committee. And we have other folks from SFBBO, from biology, and it's really wonderful to see all of you here today. So, without further ado, let me turn it over to Emily.
Thank you. Thank you, Lynn, for the introduction. And again, it's so great to see you all here. Thank you so much for coming out for my thesis presentation. It really means a lot to me. My name is Emily Moffitt, and today I am going to share my master's thesis research, which is called Using Stable Isotope Analysis to Infer Breeding Latitude and Migratory Timing in Juvenile Pacific Slope Flycatchers. So here's an outline of what will be covered in my talk today. I'll begin with an introduction on the background behind this topic. I will then discuss some of the related research in this field. I will then go over the objectives of my study, which will be followed by a brief overview of the methodologies that I used during my data collection. I will also discuss how I analyzed my data and the results. And finally, I will conclude with a brief discussion of my findings and go over some potential opportunities for future research. By the year 2050, 30 to 50% of all species may be extinct. Our planet is currently suffering from great losses in biodiversity. Bird species in particular are at risk. According to the International Union for Conservation of Nature, as of September of 2016, there have been 140 avian species that have been listed as extinct, and 218 species that have been listed as critically endangered worldwide. This is followed by many other species being listed as endangered, vulnerable, and threatened. Migratory songbirds in particular have been experiencing significant population declines. In fact, 57% of all migratory songbird species in North America are declining. For example, the wood thrush. The wood thrush is a neotropical migratory songbird, which means that it breeds in North America and winters in the tropics. And this bird has lost 50% of its total population since the 1960s, according to the Breeding Bird Survey. This decline has been demonstrated in many other species that have also been experiencing high rates of mortality, especially during migration. Now I know what you are all thinking right now, that migratory birds have it easy, vacationing every winter in the tropics and leaving the resident birds behind to shovel the snow. Well, this may come as a surprise for you, but that's a common misconception of migratory birds. Each year, migratory birds fly thousands of miles from their summer breeding grounds to their wintering grounds and then back again. They do this in search of optimal food and nesting conditions. Some of the challenges that they face during migration include exhaustion and starvation from low energy, the mental pressures associated with navigating, unfamiliar habitats and limited resources, physical land barriers such as large bodies of water and tall buildings, natural disasters, inclement weather and climate, competition from other species and predators. On top of all that, migratory birds are facing threats from human impacts such as habitat loss from urbanization and agriculture, the spread of disease and parasites, domestic and introduced predators, and quite possibly the greatest threat of all, climate change. So we know that these migratory birds are struggling, but the question is, where exactly are these birds experiencing declines? Migratory birds use flyways to travel between their breeding and wintering grounds. There are 4 major flyways in North America. Many species rely on the Pacific flyway, which is the westernmost flyway. We can't quantify avian mortality unless we can understand where these birds are breeding, where they're stopping over during migration, and where they're wintering. A concept called migratory connectivity allows us to better understand these population declines. Migratory connectivity is the study of populations by determining links between individuals in their breeding grounds, their stopover sites, and their wintering grounds. Understanding migratory connectivity allows researchers to draw conclusions about populations, specifically like the viability of a population to survive, the ability of a population to evolve with changing environments, to be able to determine how much genetic variation there currently is in a population, to determine where these populations may be exposed to diseases or parasites, and to better gauge the amount of suitable habitat that's available to meet a population's needs. Much of the research in migratory connectivity has focused on making connections between breeding and wintering grounds, but not stopover sites. Quality stopover habitats are critical to a bird's survival. When migratory birds arrive at a stopover site, their energy is already low. These birds rely on these sites to rest and refuel. They also must optimize their time spent foraging on stopover because they're facing time pressures such as to get to their wintering grounds in time to claim a territory, or to get to their breeding grounds in time to claim a nest site or a mate. Fully understanding migratory connectivity, including the identification of stopover habitats during migration, is critical to avian conservation. Past approaches to studying migratory connectivity have included bird banding, which is tracking movements of individual birds through tagging, citizen science, which is where members of the public collect data on birds, or advanced telemetry, such as radio, satellite, or GPS transmitters. Although these approaches have yielded valuable information, there are several disadvantages, such as requiring birds to be recaptured or high equipment costs. Because of these disadvantages, there have been large gaps left in the understanding of migration for many species. One species in which virtually no migratory connectivity information exists is the Pacific slope flycatcher. The Pacific slope flycatcher, which was formerly called the western flycatcher, is a small neotropical migrant songbird. Their diet consists mainly of insects, and they breed in coastal habitats with coniferous and pine oak forests. Their known breeding range stretches from southeastern Alaska to Baja California. These birds spend the winters in tropical habitats of Baja California and Mexico. The International Union for Conservation of Nature has listed the Pacific Slope flycatcher conservation status to be of least concern. Breeding bird survey trends have shown a slight decline in populations between the years of 1966 and 2012. The causes of this decline is poorly understood. There have been a few studies showing that flycatcher populations have declined in human-created clear-cut forests of northwestern California, but there have not been any other studies throughout their range. The National Audubon Society lists the flycatcher to be climate threatened, and climate model projections predict that more than half of this bird's suitable habitat will shift over the next 60 years. These predictions, in combination with the current population trends, emphasize a critical need to better understand the migratory connectivity of the species. As previously mentioned, historic methodologies in migration studies have been problematic. But there is a practical and feasible alternative, which is the analysis of intrinsic markers. Intrinsic markers occur naturally in nature and are incorporated into animal tissues after food consumption through normal metabolic processes. A sample of an animal's hair, fur, feathers, claws, or blood will contain information about the external environment in which that specific tissue was grown in. These tissues can then be analyzed for environmental contaminants and trace elements such as mercury, for mitochondrial DNA and genome sequences, for blood parasites and pathogens, and for stable isotopes. These markers, and stable isotopes in particular, have been shown to be useful in studying the migratory movements of wildlife. So, what are stable isotopes? Stable isotopes are chemical elements that vary in atomic weight but do not degrade over time. Stable isotopes of elements such as hydrogen, oxygen, carbon, nitrogen, and sulfur are naturally present in nature, and they vary geographically across different habitats, in precipitation, and amongst food webs. Because these isotopes do not degrade, signatures from the food that an animal eats in a specific location or habitat are reflected in that animal's tissues. Birds go through an annual molt cycle in which they grow new feathers in to replace the old ones. For a migratory bird, this process may occur on their breeding, stopover, or wintering grounds. So when birds migrate, the information about their previously occupied habitat is carried with them in their feathers. The analysis of stable isotopes have implications for migratory connectivity. The heavy hydrogen isotope, deuterium, varies with latitude. Deuterium in the environment will become more negative with increasingly northern latitudes. So the deuterium in bird feathers can essentially be correlated with deuterium in precipitation. Known values of deuterium in precipitation have been mapped globally by the International Atomic Energy Agency. Deuterium has been widely analyzed in connectivity research to determine the migratory origins of birds. In fact, these methods have been applied in hundreds of studies on wildlife since the late 1990s. These are just a few of the publications that have made successful inferences about the migratory origins of birds. Stable isotopes can also tell us about an animal's previous habitat, diet, and their trophic position. The heavy carbon isotope, carbon-13, will vary based on plant photosynthesis. Terrestrial C3, C4, and CAM plants are distributed geographically with climate. Carbon-13 will be more negative in C3 plants, which are found in temperate habitats with cooler and wetter climates. Carbon-13 will be more positive in C4 plants, which are typically found in semi-tropical habitats with hotter and drier climates. The heavy nitrogen isotope, nitrogen-15, will vary based on trophic levels and an animal's diet. Tissues of consumers that rely on high-protein diets will be more positive in nitrogen-15 than lower individuals in the food chain. For birds, these nitrogen isotope levels in feathers will vary with their diet, such as in diets that consist strictly of insects, or diets that consist of insects and plants such as seeds, or diets that are just strictly fruit. Nitrogen isotopes also vary with soil content, such as in regions with high agricultural activity. Many studies have analyzed the carbon and nitrogen isotopes in feathers to examine diet variation across habitats and to investigate trophic relationships. So this brings me to the research objectives of my study. The overall purpose of my study was to contribute to the assessment of migratory connectivity in Pacific slope flycatchers. Specifically, I had 3 goals. My first goal was to infer the breeding origins of birds that migrated through the South San Francisco Bay Area at different times during the fall 2014 season. My second goal was to use stable isotope analysis to examine the habitats that the flycatchers were previously exposed to on their breeding grounds. And my third goal was to see if there were any relationships between standard flycatcher measurements, physical measurements, and breeding ground characteristics. The study site for my project was the Coyote Creek Field Station, also known as CCFS. CCFS is located in Milpitas, California, within a small riparian corridor at the southernmost portion of the San Francisco Bay. CCFS is also situated in the Pacific Flyway and is operated by the nonprofit organization, the San Francisco Bay Bird Observatory. SFBBO has been monitoring wildlife at CCFS since 1987. They've been doing this through a bird banding operation that was administered by the United States Geological Survey. CCFS is an important stopover site due to its location along the Pacific Flyway and because it's located next to Coyote Creek. There's an abundance of quality riparian habitat at CCFS, and a variety of migratory bird species stop here year-round to forage. Each fall, CCFS experiences an influx of juvenile, also known as hatching year, Pacific slope flycatchers. Adult flycatchers were not considered for this study, as very few of them migrate through CCFS. This influx of young birds may be due to a coastal effect in which some of the juveniles have deviated from the rest of their population who migrate along the main inland route of the Pacific Flyway. The flycatchers typically migrate through CCFS between the months of July and November in groups of 80 to 250, which has been a consistent pattern over the past 15 years. These juvenile birds are ideal candidates for my study because they wait to molt their feathers until they reach their wintering grounds. So when the flycatchers arrive at CCFS in the fall, their feathers contain isotopic signatures from their breeding grounds. Data collection in the field was conducted at CCFS from July 20th through October 12th of 2014. Data was collected during the normal CCFS banding hours, which are 3 days a week for 5 hours each day after sunrise. Data was collected by myself and 22 other trained volunteer banders. The flycatchers were caught in mist nets, carefully extracted, banded, And then once all the data was collected, the birds were safely re-released into the wild. For each bird, I pulled 2 tail feathers for stable isotope analysis. Additionally, standard banding protocols at CCFS require taking a suite of morphological measurements on each bird. For the purpose of my study, I only used 8 of these measurements in analyses. Once the field season was completed, the flycatcher feathers that I had collected were prepared in a laboratory for stable isotope analysis. Laboratory procedures were conducted from October of 2014 through January of 2016. That was 1.5 years. The feathers were first cleaned to remove surface oils, right here, following a multi-day protocol which consisted of detergent, chloroform, and methanol rinses. These procedures were conducted under an evacuating fume hood in the protein lab of the San Jose State Biology Department. Once all the feathers had been cleaned, they were then cut into tiny little pieces and weighed to measure 0.1 to 0.2 milligrams using Dr. Alex Gershensen's analytical balance. After each sample was weighed, each feather sample was weighed, they were carefully encapsulated into very small tin and silver capsules. I prepared a total of 374 samples for stable isotope analysis. This number includes several replicate samples to measure instrument accuracy. Once all the samples had been encapsulated, they were sent to the UC Davis Stable Isotope Facility, where they were analyzed using isotope ratio mass spectrometry. In preparation for data analysis, I had to obtain some secondary data as well. I obtained 2 shapefiles. The first, which is the graphic on the left, is a map of the average deuterium in precipitation across North America. This map was created by researchers using data from the International Atomic Energy Agency. We call this map a precipitation isoscape, and it serves as the foundation for essentially placing the feather deuterium values. To improve breeding origin results, I adjusted this isoscape for the foraging guild that the Pacific slope flycatchers belong to. The second shapefile that I obtained, which is the graphic on the right, it was a map of the known geographic range for Pacific slope flycatchers. This map was used to further the map was used to further improve the assignment results by constraining origins to regions only within this map. So, how did I analyze all this data? To address my first research objective and infer the breeding origins of the flycatchers that migrated through CCFS at different times during the fall season, I ran spatially explicit likelihood-based assignment models using R scripts from researchers who have conducted similar studies. This is basically just a fancy name for an analysis that follows a Bayesian framework by using probabilities to assign birds to likely versus unlikely origins at 2-to-1 odds. I tried out 2 different approaches using these models. The first approach, which resulted in 1 map, examined the likely origins of the flycatchers collectively as a group. The second approach, which resulted in 13 different maps, examined the likely origins of the birds on a more individual level. For this approach, samples were organized by the 2014 calendar week that the birds were captured and analyzed separately. To support the 13 weekly assignments, I conducted a linear regression of all 169 samples to see if there was a significant relationship between the date of capture and feather deuterium. To address my second research objective and examined the habitats that the flycatchers had been previously exposed to on their breeding grounds, I ran a k-means cluster analysis of all 3 isotopes. I paired this with a multivariate analysis of variance to see if there were significant differences between the clusters. And finally, to meet my last objective and examine the relationships between morphological measurements and breeding origin characteristics, I conducted a canonical correlation analysis of the 2 different multivariable data sets. The independent variable data set contained the 3 isotopes, and the dependent data set contained the 8 morphological variables. For all of these analyses, any conditions that were required were met prior to analysis. Now, for the part that you've all been waiting for, the results. The group assignment model was able to assign 96 out of the total 169 birds to probable breeding origins throughout the known Pacific slope flycatcher breeding range. The legend scale over here specifies the proportion of birds, which was in this case over half of the entire sample, that had consistent feather deuterium with pixels on this map, based on the chances of the assignment being a true origin at 2-to-1 odds. I interpret this to showing that the most likely breeding origins for the majority, which is shown in the darkest blue color, were regions in northwestern California, throughout western Oregon, and in southwestern Washington. This map also shows a secondary peak in assignments, which are the colors in that dark green shade, in which a second group of migrants were likely associated with regions in northern Washington and the southern British Columbia border. My primary research objective was not only to determine the breeding origins of the flycatchers as a group, but to investigate their origins in smaller groups of birds that migrated through CCFS at different times during the season. The linear regression showed a significant but weak correlation, negative correlation between capture date and feather deuterium. This regression implies that birds from more northern latitudes arrived later in the season than birds from more southern latitudes. This regression, paired with the 13 weekly spatial assignments, gave evidence for there to be potential distinct groups of populations moving through CCFS at different times. The Gantt chart on the right further breaks down this trend, with a timeline of when the suggested populations move through CCFS. Again, these distinct regions were inferred from the 13 weekly spatial assignment maps. To further support these findings, I examined how flycatcher feather deuterium changed throughout the season, and I found there to be an average shift of negative 2.9 parts per million each week. Here's an example of the weekly assignment models that I conducted. The map on the top displays the most likely breeding origins for the birds that migrated through CCFS early in the season, which was during the 4th week of data collection. This model correctly assigned 3 out of the total 4 birds from this week to regions within the coastal California mountain ranges. In comparison, the map on the bottom displays the most likely breeding origins for the birds that migrated later in the season during the during the 11th week of data collection. These regions were around the British Columbia border in northern Washington as well. Moving on to my second objective, which was to examine the habitats of the flycatchers on their breeding grounds. The k-means cluster analysis showed a natural clustering of 2 isotopically distinct groups amongst the individual flycatchers. This result was further supported by the MANOVA, which showed that there were significant differences between the clusters. What I've interpreted this analysis to show is that the birds that were classified in cluster group number 1, which are the numbers in the red, on average displayed more negative deuterium, more negative carbon-13, and more negative nitrogen-15, which this could suggest that based on lower deuterium, as we know, these birds associated with more northern regions. Based on the lower carbon-13, these regions may have been in cooler and wetter climates. And based on lower nitrogen-15, it's possible that these birds from regions may have foraged for insects paired with vegetation. Alternatively, the birds in cluster group number 2 on average had higher values for all 3 isotopes, implying that these birds associated with more southern, warmer, and drier climates and possibly had some sources of fruit in their diets. And to address my third and final research objective, which was to examine the relationships between morphological measurements of the birds and their respective breeding origins, I hypothesized that there would be no relationships between the 2 different data sets. Prior to analysis, I found that one variable, body weight, was asymmetrically skewed to the right. So a log transformation was performed for correction. All of the variables within their respective data sets were independently correlated from one another. And the results returned one statistically significant canonical variate. Root score calculations returned significant canonical loadings for the following variables, ranked by the weight of their contribution to the analysis. Deuterium was weighted the highest, followed by the log of weights, carbon-13, fat score, and weighted the lowest was nitrogen-15. I interpret this analysis to show that birds with lower deuterium, carbon-13, and nitrogen-15 from more northern, cooler, and wetter regions typically displayed lower body weights and lower fat scores. So, to summarize this all, my analysis of stable isotopes in the feathers of the juvenile Pacific slope flycatchers that migrated through CCFS at different times during the fall of 2014 has allowed me to speculate that these birds most likely originated from throughout their known breeding range, that juvenile flycatchers that migrate through CCFS come from distinct geographic populations, that these birds also possibly utilize a chain migration strategy in which they migrate in a sequence with the southern populations migrating first, followed by the northern populations. These birds potentially occupy different habitats on their breeding grounds. And finally, my analyses suggested that certain morphological measurements may vary in individuals from different habitats or from different latitudes. The approach to examining potential flycatcher origins on a smaller scale versus examining the origins of the group as a whole has led me to speculate that these birds migrate from distinct regions. The weekly assignments provided us with new information about the population dynamics of this species. Most importantly, my findings suggest that these juvenile flycatchers who breed in multiple regions of North America rely heavily on migratory stopover sites such as the Coyote Creek Field Station. The influence of temporal data to explain variation in deuterium of the bird feathers has been investigated in several other studies with similarly weak regressions. These researchers have also found evidence for potential chain migration in other species, but we cannot fully confirm that the flycatchers apply this strategy unless we know exactly where they are wintering. Similar conclusions have been made about migratory timing based on weekly assignments with smaller sample sizes. This has been investigated in several other studies using stable isotopes and also genetic analyses. My examination of breeding ground habitats suggested that the flycatchers associated with two different climate types. Some birds were associated with more northern regions in cooler and wetter climates, and they were possibly exposed to habitats with more plants and seeds for foraging, in addition to their normal insect diet. Alternatively, other birds associated with more southern regions in warmer, drier climates, where there were potentially more opportunities for them to forage on fruit rather than seeds. My interpretation of certain birds originating from cooler and wetter climates is supported by research showing that carbon-13 varies in habitats with temperature and moisture influences. It is known that habitats within the Pacific Coast mountain ranges are characterized by climatic moisture scales, which vary with latitude. The cooler, wetter regions are found farther north. My speculation that birds had different foraging opportunities is supported by similar multi-isotope studies that have linked variation in carbon-13 and nitrogen-15 to diet. Studies have also found that proximity to agricultural operations will highly influence nitrogen-15, so more research would be needed to really draw better conclusions on flycatcher diet. And finally, my examination of morphological variation in relation to breeding origins has suggested that some of the flycatchers associated with more northern regions and showed lower body weights, while other birds associated with more southern regions and showed higher body weights. The trends that we had been seeing with the, The trends that we have been seeing with the habitat characteristics remain consistent with these findings. The interpretation of these trends in body weight and fat score have potential implications for, now we go to, migration distance, in which studies have found that some birds who migrate longer distances have lower body masses. But this could also do with clinal variation, which research has shown that is typical for traits in animals to change with geographic range across latitudinal gradients. This possibly could have to do with environmental stress or malnourishment, where studies with captive birds have shown that body mass and fat score tend to be lower due to stressful environmental conditions. Overall, again, more research would need to be done to draw proper conclusions about these relationships. So, in summary, I'm going to conclude with 3 different takeaway points. The first is that the analysis of stable isotopes has allowed me to meet the objectives of my study and make basic inferences about the probable geographic origins and habitats of migrating juvenile Pacific slope flycatchers. More research using advanced methodologies is needed to better understand migratory connectivity in Pacific slope flycatchers. That being said, the findings from this study provide new insight on the flycatcher migration and lay a foundation for future research and management. It is my hope that wildlife researchers and managers can now better define migration patterns and breeding territories in Pacific slope flycatchers, better explore migratory connections between flycatcher populations, and subsequently, better track population decline. This research can help managers better understand the importance of stopover ecology in migration and better understand this species' ability to adapt to a changing environment, especially with climate change. This research could also help improve better tracking of the future spread of disease and parasites. Finally, the application of temporal methodologies and investigating migratory timing in this study demonstrates a potential new ability for researchers to explore migratory movements on an individual level while making population-level inferences. So with that, I have several recommendations for future research. I suggest expanding upon this study to further assess migratory connectivity in flycatchers by targeting additional juvenile birds and adult birds in other stopover locations along the Pacific Flyway. I would also recommend sampling birds during the spring that are coming back from their wintering grounds to potentially determine where they were spending the winter. These improvements would allow us to make inferences about migratory connectivity from all aspects of the flycatcher population. My ability to make inferences about breeding ground habitats and diet was limited by the fact that I did not have known carbon and nitrogen isotope data from potential regions that the flycatchers could have occupied. So my idea to improve upon this would be to sample vegetation and insects for stable isotope analysis from throughout the known flycatcher breeding range. Doing this would serve as a basis for comparison to better examine habitats and diet using stable isotopes. As mentioned, stable isotope data alone has laid a foundation for further building upon our results. We could incorporate additional secondary data resources to further refine breeding origins of the birds. For example, one idea would be to investigate the possibility for the flycatchers at CCFS to have originated from eastern breeding territories of a very closely related species, the Cordilleran flycatcher. Another idea would be to obtain flycatcher feathers from known origins, either through museum specimens or other banding stations, to better calibrate the precipitation isoscape. Lastly, we could also try to analyze banding and encounter data from the bird banding laboratory to better estimate directions of migratory movements. And finally, to build upon my results, I would look into incorporating newer technologies such as genetic analyses to determine genetic differences between populations, to possibly deploy light-level geolocation devices for seeing very specific movements, or conducting compound-specific stable isotope analysis for a more specific evaluation of habitat and diet. And here are my references, and I'd just like to take a moment to acknowledge all of the people that have supported me throughout these past few years. Dr. Lynn Trulio, my thesis advisor, you have given me amazing guidance these past few years, and I would not have been able to do this without you. Dr. Kristen Rugg, it's very late for you right now. Thank you so much for being here and all of your advice, and as well Dr. Scott Schaefer, my committee members. A huge thank you to the filming guy back there in the corner, my boyfriend Justin, and our roommates, our cats. They've all been incredibly patient and supportive through all of this.
Very helpful.
Yes, that's my actual thesis that he's messing up. Thank you to all my funders, the College of Social Sciences, my Indiegogo crowdfunding supporters, Isoprime and Green Mountain Antibodies. I also want to thank everyone at SFBBO and at CCFS for providing me so much help during data collection and giving me a place to actually do this research. Especially Josh Scullin, you've been so amazing through all of this. Thank you to everyone that supported me through my laboratory analyses by either giving me space or helping me encapsulate all those feathers. And a huge, huge thank you to all the people that helped me figure out essentially how to make these assignment models. Tim Walker and Stephen Van Wilgenberg from Environmental Canada. I would have not have been able to do this without them. Thank you to my cohort, everyone, my peers at SJSU, my family, friends, and coworkers, and the birds, of course, because I wouldn't be able to do this without them. And at this point, I'd be happy to take anyone's questions.
Okay, before we start with questions, hi Kristen. Proper introduction of Kristen Rugg. She is a PhD researcher with UC Santa Cruz and UCLA.
That's
UCLA,
right, yeah.
yes. And she has been extremely instrumental in helping Kristen through her thesis work. So to begin with, if the thesis committee has any questions, we'll start with maybe Kristen. If
Yes, so you
have
don't have to.
any questions?
Sure, yeah, I have a couple. I have sort of one technical and one bigger picture that came up while I was listening to your talk.
Okay.
Really nice job, by
Thank
the
you.
way. I really enjoyed seeing it all come together. It was just wonderful. So the first one was, in the k-means clustering, do you have any sense as to which of the isotope variables is driving that pattern? Was there any kind of loading score or anything that could help you differentiate whether those clusters were mostly due to the nitrogen, carbon, or hydrogen?
No. So the question was I should probably play from this slide. Was about the k-means cluster analysis that I ran, and if there was a certain if one of the isotopes, like the canonical correlation, was really driving this correlation. And from what I understood of the cluster analysis, that from interpreting it the way I could, I did not see a way to get that data. I know an option would have been for me to run an MDS, multi-dimensional scaling of these to see exactly how the clusters are related and why, but I did not do that for this analysis. But I have a feeling it was deuterium that was driving the biggest separation. That would be my That would be my
guess too.
Right.
It would maybe help you, if you knew that, it would help you determine how much of this could be sort of diet related.
Right.
Versus, yeah. So that's interesting. Okay, and then the other one is sort of like a big picture question. So let's say somebody was going to give you $100,000 tomorrow to help with, Pacific slope flycatcher conservation, and they wanted you to inform that, how you're going to spend that money based on your results. What would you recommend based on what you found you would do with that $100,000 to help save these birds?
That's a very important question. The question was, what would I do if someone gave me $100,000 to further protect these birds, the Pacific slope flycatchers, and support their populations and the species? So that's a good question, because my first thought is that I don't know how to properly support a population or support an area unless we know where these populations are declining. And so I know one of the biggest things is that a lot of not is known about the wintering origins of these birds. So I would selfishly like to take that money and do another project to further study them rather than put that money into restoring a specific habitat, because I don't actually know if that habitat is causing the decline or not. So I would take that money to continue researching the birds and then use that to make further recommendations on where to protect them.
Excellent. All right, that's it from me.
Okay, thank you, Kristen.
Can you cover our salaries on that?
Yeah, all my field researchers.
Zach, did you have a question?
I can't remember if we discussed this when you were looking at the stable isotope analysis, but did you think at all about sectioning the feathers and then looking at individual sections so that you might be able to infer a timeline associated with how long they stop? Or
Yes, we did talk about that, and actually you set me up with Hillary Young to talk about that as well, and I had some great phone calls with her, and I got really excited about the idea of doing that. I even had that on one of my slides as future research, but it was...
I told her to take it out.
But so for this study, actually, to give you an idea, UC Davis required 0.1 to 0.2 milligrams of feather, which in the flycatcher feathers amounted to close to a quarter of or half of an entire feather. So if I'd have to find another lab that could use smaller sizes to do something like that, but that would be super interesting, and I would love to look into doing something like that in the future. So.
Okay.
Yep, Lynn?
So Emily, we know that they come to the riparian station it's a stopover site. We know that the more northerly birds are, maybe their condition is not as good as the more southerly birds, right? They don't weigh as much.
Right. Mm-hmm.
So, two questions. One, do you think the birds up north are migrating later because they breed later? And because these are young of the year.
Yep.
So, do you think that's kind of a tangential question. The real question is, what can Coyote Creek Riparian Station do for the birds who, especially the northerly birds who are coming through and may need to tank up? You know, we know when they're coming through. So what could be done for the birds at the riparian station?
That's a very good question. So another thing about these birds, which I mentioned in the beginning, let's see if I can get back there. I will. So there's research that's shown that, no, that's the wrong slide. Well, I can't find the slide, but that these juvenile birds that migrate through, and when you don't see adult birds, it's they're kind of they're taking these birds. Not all the juvenile birds have deviated from the rest of the population that migrate on the main Pacific flyway inland route. So juvenile flycatchers also migrate inland, but we're seeing the birds that have deviated from the rest of the population migrating through CCFS. And with that, research has shown that these are the birds that are likely not going to make it that next year, because they're not following the same route as the rest of the birds. They just, for many, I'd have to look at more research, but these birds might not make it. Flycatchers, as all of us banders know, are incredibly stressy birds sometimes to band, and we take a ton of measurements on them, which are important measurements to take. But I'm wondering if, at least from a bander's perspective, if we can, during those times in the year that they're coming through from northern regions, if we can perhaps not take so many measurements to stress them out, because we don't know where they're going from there. We do know that they continuously rely on this habitat, and I think SFBBO does a great job at has had a great habitat for these birds to forage upon. But I'd say minimizing the stress from the birds that are coming through at the end of the season, especially we saw these birds based on the isotope analysis from potential regions close to Canada coming through only during the last 2 weeks of the season. So whereas other birds would sort of trickle through, could mean many things, but maybe as banders, which it's shown that we add extra stress by banding them. I think putting a band would be great, but maybe not taking so many measurements to stress them out if they're showing signs of stress, or, yeah, be my main suggestion.
Okay, questions from the group?
Actually, just
Yep.
Emily, on that topic, so from what I got from the presentation is that some animals were lean, they say, and others were more robust.
Yep.
You were able to kind of give a little correlation there. So is this to say, though, especially with this recent discussion that you just brought up, that the animals actually then had the leaner animals actually had a poor body score? Is that what I'm hearing? Because I figured the lean animals were doing great and the more robust ones were particularly happy. So that was kind of what I understood. Is it
Right.
really your interpretation that the animals were scored poorly?
No. So that type of measurement, body size, which Scott, you've done on many of your birds to wing size, and it's body size measurement, which you measure the wing length divided by the weight of the body. That gives you body size, as well as there are other measurements you can take that actually show if stress or poor quality food has been consumed. But the lower weights and lower fat scores although they showed significance in the analysis, we'd have to farther look into how different they were. I think they might have just been, it could have just been birds that were lower in weight, not necessarily that they were physically in a poorer condition. Body weight and fat score alone don't tell us the type of condition that the bird's in.
That's correct.
Thank you.
Good question.
Oh.
Birds.
Scott, do you have a...
You know, one thing you could think about in the future is if you took a sample of the feathers, and you'd have to figure out which feathers to take, but you
Right.
can look at court levels and things like that.
Yeah.
That might give you an indication of stress level in relation to body size and mass and stuff like that.
So you can do that just from feather? You don't need blood or...
No, you can. I think some studies have shown now, more recently, that you can measure corticosterone from the feathers. What I don't know is if it takes a lot of feathers, or you know, if it's dependent on how much feathers you put in there. But I know it's starting to become, you know, something that people are looking at.
Yeah.
From the tissue at the end of the feather?
No, I think it's a signature that's actually laid down at the time the bird was molting.
Yes, Dr. O'Malley.
Just because I'm mildly obsessive, the K
Oh,
cluster,
sorry. Yes.
graph, I wondered.
Let me find it. I have a lot of animations to go through.
So I was just trying to understand. It looks to me like the means are separated for the deuterium, and they're not really separated for the carbon or the nitrogen. Is that
Right.
not reflective of possibly driving it, or?
Yeah, so I did look in, so that was separately, I looked at how the clusters were partitioned, and based on means alone, so I think, and I further checked, and the difference between the hydrogen, I believe, was a 17% difference between the values.
99 to 70 there.
Yeah, yeah, so carbon clearly means not much. And then, but also the nitrogen was a 15% difference, so.
Right, but if you look at the overlaps there with more, you know, really with the variability, it looks like it's, I mean, I was just kind of going back to that original question that you had, and I was looking at the graph and I was saying, well, it looks to me like the deuterium is, because it's sort of, they're separated on that axis. And I understand that there's, you know, statistically, you don't have a number saying that's what it is.
Right.
But sort of descriptively, it looks like the deuterium, you could make a case that
Deuterium explained was.
I mean, if I'm looking at those clusters, I see this horizontal shift that's bigger, certainly, than the vertical shift, and
Right.
kind of hard, of course, to read going
Sure.
back. But anyway, that was the whole thing.
Yeah, and that's, again, I do believe that deuterium did explain most of it. I guess because like the canonical correlation, I had something to tell me, like a number-wise, but I guess that you can just say that deuterium did, and you know, throughout this whole thing, deuterium's been the real answer in this study, I think further supports the cluster analysis. So
The other thing that I noticed that you had said that the folks at Petty Creek are a little bit separated from the main migration group, right?
yeah,
Do you think that makes them more vulnerable to climate change, or more vulnerable or more interesting genetically because of that potential speciation happening? How long do you think it's been? Any speculation on that relationship?
yeah. I wish I had a map to show like the routes of the different routes that birds take with the flyway, but well, it's because in other species, we see different age groups coming through, and you know, I think it's when you see in this situation just a ton of young birds that have come through. Those young birds have deviated from the rest of the population. It's not that young birds take this route; it's just this specific group has. I don't know. I'm not really sure why, or if it's just these birds that are young and they don't know, or if they're, you know, forced to migrate early earlier than adults or later than adults, that type of thing. It's their first year. But that would be really interesting to further understand where they're coming from and where the rest of the population's coming from, which is why I'd really love to move inland and study the rest of the population and see how it compares to these birds. So,
Good study.
thank you. All right. Yes. Hi, Jay.
Great talk.
Thank you.
And I again am drawn to this graph.
Okay.
It is visually stunning, a separation there. And I'm thinking to the range map too of how you've grouped your northern versus southern breeding things. Is there some sort of geographic split, elevation split that is, driving the deuterium off of climate that is, what is clustering the northern to the southern?
That's a good question. And so, let's see. So if we look at the main group assignment that I did, which was all the birds at once, this assignment told us that the majority of the birds were associated with this grouping which means that the deuterium in these ranges is the same. So and then in the deuterium maps, you'll see these contours of how the deuterium changes. So it's different here. This area is all the same. It's different here. It's different there. It's different there. It's possible that these groups of birds are separated from, in this area, in the blue, have a big separation from the ones from up here, which we showed. The assignment said that there were two different groups. In the individual maps, we did see a lot of birds, a lot of the maps showing that for even the smaller samples by week. But we wouldn't know, but the timing, when you come in and look at it further with timing, then you would see the birds coming from the farther top. So I don't know why they're separated, why the populations are separated, but I mean, we know that these birds breed as far north as southeastern Alaska, so, but none of the birds in this study came from that area, but.
Okay, so at this point, the committee needs to convene
Okay.
and talk a little bit. So, and see if we feel that Emily is ready to move on, which, and so we're going to do that. And I guess can I just?
Yes, of course.
Let me take this. So we'll be back in just a couple minutes. I don't know if there's any goodies back there. You can
Yes,
have a
help
few,
yourselves.
and we'll be right back. Hi, Kristen.
Can answer. All right.